Good morning, happy summer, and welcome back to FirstThought Focus. FirstThought is a life sciences focused expert network with a mission to bring subject matter expertise back into the expert network industry. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts and clients, while also covering a wide range of topics that are relevant and important to our space. You can find the replays of our past events and conference recaps on our website, firstthought.io/focus. Today, we are thrilled to welcome physician scientist and corporate strategist, Dr. Frank David, to discuss his new book, The Pharmagellan Guide to Analyzing Biotech Clinical Trials. Before we get started, I do want to remind everyone listening that we will be taking questions from the audience throughout today's discussion. If you'd like to ask a question, please submit it via email to hello@firstthought.io. Today, we're doing something a bit special. For the first 5 people who send in questions to hello@firstthought.io, and they need to be good questions, we will be sending you a free copy of Frank's new book. I will be in touch with those lucky listeners after the event later today. And with that, I'd like to turn things over to our moderator, Neil Canavan.
Hi, and again, good morning. My name is Neil Canavan. I'm the Managing Director of the KOL Network here at LifeSci, and the moderator for today's discussion on the new book by Frank David, Pharmagellan Guide to Analyzing Biotech Clinical Trials. Now, this will soon be a TV movie, but I hope you stay with us. Playing the part of Mr. David will be none other than the slow-talking, fast-driving Vin Diesel. Discussing this casting choice with me today is the author himself, Mr. Frank David. Frank, welcome to the webcast.
Thanks for having me.
With Matt Damon. All right, first off, Vin Diesel, personally, I would have gone with Matt Damon, but I digress. Let's move on to the topic at hand. These are the ins and outs of analyzing clinical trials. Now, listeners, the way this is going to play out is I'm going to ask things that are covered in the book that make me feel stupid and helpless. And the reason they make me feel stupid and helpless is that even though I have an advanced science degree, I'm mostly a journalist by training. I'm not an analyst. So there's a lot of stuff I had no choice but to sign off on because I simply don't know better. The presenter says the trial's a success. It's got a p-value of less than 0.05. I write that down. The next day, the stock goes down 10%. I don't know why. So to repeat, I'm going to be asking about things that I want to know about. You, the listener, might have other concerns. So starting right now, you can dial in your questions. I will fold them in as the conversation progresses. In general, we will cover statistics like p-values, confidence intervals, the ins and outs of where to physically do a trial, as if that matters, press release shenanigans, a favorite topic of mine, how to look at patient population, trial design, subgroup analysis. There's obviously a lot of ground to cover. So as I said, dial in your questions now. And let's take it away with the first question, which is, David, why did the word underpowered piss you off? And what did you do about it?
Awesome. Well, thanks again. Just as a preview, or just as a prelude to all of this, I am a full-time consultant. We are going to talk about individual companies. You should assume that any company that we mention is a potential client of mine. If there's any direct conflicts, I will let you know. With that said, I don't remember if I said this in the book or not, but I certainly have told other people that one of the main impetuses for writing the book was that I realized I was throwing around the term underpowered in client meetings, and I wasn't exactly sure how to describe it and what it actually meant. And I actually think that, what you described as being someone with a science background and being a journalist, like that's sort of the intended audience here. It's certainly not for statisticians. It's not intended. I'm not a statistician. I'm not someone who designs clinical trials for a living. I'm really sort of in the consumer category of clinical trials. So, I think what the underpowered, the problem with the term underpowered to me is I never really understood what's the difference between just saying a trial is underpowered versus it just failed. And it felt like pretty much every time a trial failed, someone was always claiming, well, it was just underpowered. I think, the key concept there actually is that, powering is about what size effect you're actually trying to detect. So, there's this sort of triangular relationship between, the power, the minimum difference you can detect, and the size of the study. And essentially, when a trial is designed, there's a decision made about what size. Either one of two things happens. Either the people organizing the trial decide this is the size difference I want to be able to detect, and then they figure out how many patients they need for that. Or they say, this is the number of patients I can afford, and that then backs them into, this is the minimum amount of difference that I can detect. So, I think often when you see this underpowered question, it comes up, in phase 2 studies, where that second category of decision-making often happens, right? It's like, this is how many patients we can afford to put into our phase 2. So therefore, we are now powered to detect a certain difference. And I think the question that then ends up coming up when a trial like that doesn't hit statistical significance is whether that matters or not, whether that difference was too aggressive or appropriately aggressive. So if you're, that's obviously clinical context dependent. So if I'm in a cancer study, for example, looking at overall survival, and I'm powered to detect a 9-month improvement in overall survival, and then the trial doesn't pass that bar, is not statistically significant, to say it's underpowered is basically to say, yes, it was powered for 9 months, but actually 5 months would have been clinically meaningful to me. So therefore, the trial couldn't detect that smaller number. If we would have had more patients and we could have detected a 5-month difference, then that would have actually been meaningful. On the other hand, there are other situations where you might say that, again, I'll use the same example. Let's say the trial was powered for a 5-month improvement, and you say, well, actually 5 months is really where I would set the threshold for something that's clinically meaningful. Then if the trial doesn't fail, it wasn't underpowered, it was just a failed trial, because basically you've decided that any improvement less than 5 months would not have been clinically important. So therefore, that's a failed trial. So that's really that, I think that's one of the things, answering the question when a company issues a press release or makes some sort of public statement, or when analysts say that a trial was underpowered, it really requires one to look at what difference it was powered for and decide whether you think that that's the meaningful difference, or whether a smaller advantage actually would have been clinically meaningful.
You mentioned finances might be pushing this decision. I'm trying to figure out where the fault lies. Would you find this a more common issue with smaller biotechs who simply don't have the money?
It's funny. I mean, obviously I have clients that are both in biotech and in pharma, and I've worked internally at a large pharma company. I think there's always this, there's this idea out there among people who have not worked in large companies that large companies are just rolling in dough and there are never capital constraints on how to run a trial. And I think anyone who's ever been on a project team and had to argue for, had to lobby for funding and had to go in front of a decision-making group and sort of get money to run a trial knows that that's just not true. So I think you see quote unquote underpowered trials, or trials where the sample size is driven by the finances. I think you see that in large companies and you see that in small companies. However, I will say that at phase 3, certainly that's a place where usually in a large pharma company, if the decision is made to go for phase 3, I would say it's less common to see a quote unquote undersized trial. And in fact, if anything, you more often will see overpowering in large companies than small companies, by which I mean to say, you've decided that 5 months would be the improvement that would be clinically significant, but actually the the trial is so big that it can detect a 2.5-month increase improvement. And so therefore you can sort of win on statistical significance, even though that's not clinically significant. That's something that I think somewhat more often happens in large companies than in small companies. But in earlier stages, I think it happens in both.
Okay, there are a couple questions in chat already. I'm not going to address them this moment because I'm thinking we're going to cover both of these just in the course of the discussion. But questioners, yes, I will absolutely get to these questions. Now I want to turn to the very first graphic that I have. I stumbled across an article recently. I didn't get to read it because it's got a paywall, but I think that the point is pretty well taken there. And it says, this was never intended as a use of like a verdict, yes, guilty, not guilty. It was supposed to be a guide. What the hell happened? What, you know?
Yeah, it is a great, I mean, the whole history of p-values is super interesting. You know, and it has been basically just a, you know, it's turned into a convention that p less than 0.05 is viewed as quote unquote significant. And I think more importantly, not only is a convention in most people's heads, but you know, more importantly from a regulatory point of view, I mean, FDA guidance has clearly sort of relied on a p less than 0.05 as sort of their benchmark for what they consider a positive trial. So I guess what's important to know about that is, you know, many people think that a p-value of 0.05 means that there's a 5% chance that the trial is a false positive, quote unquote false positive. It's not exactly true. The strict formal definition is that there's a 5% chance that if you ran that exact trial, that you would have seen the difference as big as what you saw, even if there were no difference between the two arms. So in practice, you know, why does it matter? You know, why does that p-value matter? Why does the p-value matter? I think it matters because it gives you a sense of sort of how confident you should be that the observed difference is really a true reflects an intrinsic difference between the two arms. So
does the phrase highly statistically significant have any meaning at all?
No. So, I mean, that's just sort of, in fact, there are all these jokes out there on the internet, you know, where people talk about, you know, people comparing sort of different p-values and putting different words next to them, like almost statistically significant, very statistically, extremely, and none of it matters, right? Like, so that's one thing. And the other thing to note is that, you know, when you're outside of a phase 3 trial, you know, where there's regulatory considerations, you know, there's a great paper that was published in the New England Journal, which is titled something like "The Trial Was Not Significant, Now What?" So basically it says, okay, so let's say you have a p-value of 0.06. What does that actually mean? Okay, so again, if you're in a registration enabling trial, that has very clear ramifications because, you know, only under very special circumstances will FDA really, you know, approve something that doesn't meet the PO of 0.05 threshold. But if you're in more of an exploratory stage where you're to really where the point of the phase 2 trial is really to decide whether to invest or not in the larger follow-up study, you know, maybe you would say, okay, 0.05, 0.06, like it's still basically pointing in the right direction. And getting back to the discussion we just had about about power, you might say, okay, maybe actually what we learned here is that the difference between these two arms is less than we thought it was, but now we have more information and now we could design a successful phase 3 study.
Okay, you just used the phrase pointing towards. What I read is trending towards significance, right, which to me always sounds like crap.
Well, and again, I think in a study that's appropriate, it really comes down to the whole question of powering that you started with, right? If the trial was appropriately powered for a difference that really is at the minimum level of what you would accept as significant, then again, that's not an underpowered study. It's just a negative study. You designed it thinking you'd get a P of 0.05 and you didn't. But there are situations in which, you know, trending towards statistical significance is relevant. And I think it's most relevant in the situation where you're using, again, I'll come back to it, where you're using a phase 2 study to decide, should I invest in a further study and what should that further study look like? So you don't want to throw good money after bad money, so to speak. You know, it would be a mistake in that situation to just rinse and repeat and run the same study over again. On the other hand, from a Bayesian point of view, you know, you have learned something from your P of 0.06 study. And especially if you put that together with other pieces of evidence, like maybe, you know, you were 0.06, p-value of 0.06 on the primary endpoint, but then you also looked at a bunch of other biomarkers and other things, and they were all supportive of the underlying scientific hypothesis. You know, you might feel like, look, you've de-risked the underlying science and you've learned something new about what the true effect size really is. Maybe you've also learned something about how to better select the patients. And those could all inform a good design of a phase 3 study. So I think it's
we're getting a lot of questions here, and a couple of them are related exactly to what we're doing right now. So this refers to hazard ratio. I was going to ask about that, but I'm going to go ahead and read this question. Why is the hazard ratio emphasized more by some companies for certain trials? What kinds of patient populations and therapies are most relevant for using hazard ratios?
Good. So hazard ratios really come into play with what's called time to event studies. So, that's the most common place where you'll see them in biopharma trials. So that's a trial where sort of like a typical, many people have seen Kaplan-Meier curves for cancer, and I know you have one that maybe we'll look at later. Also in cardiology is another place where you see this a lot, where if you think about, for example, the trials of lipid-lowering drugs, where you're looking at sort of the risk of having a cardiovascular death or stroke. So what you're doing is you're basically marching out over time, and you're looking at whether an event happens or not over time. And then the hazard ratio is really the way that it's the main way that you summarize that finding. And you basically say, what is the relative chance of having the event on the drug versus in the control arm? So it's not like you can take just any old study and make a hazard ratio out of it. It would not be, if you're looking at a single parameter and you're just saying, did that parameter, what percent did that parameter decrease by, then the hazard ratio is not really the appropriate measure. Hazard ratios really are used in biopharma, specifically in the context of these what's called time to event studies.
All right, I want to go to one more math question. If I can have the second graphic up here. Confidence intervals. Now, I tried to get through this. I really did, because you're like, oh, you don't need to be a math expert for this. And I'm like, oh, man. So the question right at the bottom, do I have to be a statistician to know whether or not a confidence interval is bullshit?
Okay, great question. So, notwithstanding the fact that you defaced my lovely book. So I would say there are a couple things about confidence intervals. So confidence intervals and p-values are somewhat complementary to each other, right? So a p-value is really looking at the point estimate for each of the arms and saying, how likely is this that I could have seen this big a difference between the 2 point estimates if the two arms were actually the same, there were no difference between them. That's p-values. Confidence interval asks something a little bit different, which has to do with, for each of those point estimates, whether it's in the control arm or whether it's in the experimental arm, how much, how confident am I? If I ran this experiment over and over again, where would 95% of the answers lie? For a reader of clinical trials, I think there are two, non-mathy things to note about that. So the first non-mathy thing to note is that if the confidence intervals do not overlap at all, then the p-value is, I think, always, from a mathematical point of view, less than 0.05. But you can have situations where the confidence intervals overlap a little bit, and that can still be quote unquote statistically significant at a p-value of 0.05. We're not going to get into the math there, but suffice it to say that that can happen. So I think when the p-values are very separate from each other, that's obviously a very good sign. When they're close, usually that correlates with these situations like p-values of 0.03, 0.04, and it's kind of complementary. I would say the other reason why confidence intervals can be useful in some situations is just giving you a sense of really how big or small the spread is in effect. So I think one of the examples I gave in the book was a trial that was looking at exercise tolerance. And it was, I think it was looking at, and the confidence interval around the sort of improvement in the ability to do one of these exercise tests was between, 1 minute worse to 5 minutes better or something like that. So there was some tolerance around the confidence interval. So I think that in some clinical situations that can help you figure out even if, regardless of whether the study is quote unquote positive or negative, it can tell you really where the bulk of the action is, right? So if the confidence interval is very wide, it can, even if the study is significantly significant, it means that for any individual patient, there's a pretty wide variability in terms of where you think they might land. Whereas when the confidence interval is very narrow, it tells you that, that there's a lot less uncertainty on an individual patient-to-patient level. I will tell you that for me, as someone sort of on the consulting who advises companies and advises investors, I would say that I find confidence intervals maybe a little bit less important than I would if I were actually treating patients. I think that if you're actually, physicians who actually sit in front of patients, confidence intervals can be extremely useful in spelling out and sort of contextualizing how likely are you, Neil, to see a benefit from this drug that we're going to give you, and what's sort of the range of benefits. I think for people like us, it can be complementary. Certainly there are clinical situations in which certainly if you're trying to assess the commercial prospects for a drug, you might be very interested in sort of answering this question of, okay, what are clinicians likely to think about these confidence intervals? So I'd say that it happens sometimes, but usually probably not something to get wrapped around the axle over.
Okay, I'm going to move on now, and I'm going to go off script because I'm getting a lot of questions. Yeah. So we're going to be jumping around all over the place. This one is, I'm going to try to keep it within a flow of thought here, and this does go to what you analyze. This one goes to intent to treat versus evaluable and dropouts and stuff like that. I just want to drop in one quick anecdote. I remember at ASH one time, they put up the discontinuation slides, and one of the reasons there was, I don't know, 100 people in the trial. 2 of them were discontinued because they committed suicide. 1 of them was murdered. I'm like, okay, yeah, boy, we dropped that one out of the analysis. So here's the question. One thing in clinical trials presentations that drive me crazy is the concept of last observation carried forward, which to my understanding is that if you lose a patient for a clinical trial, you carry their last data point forward for accounting in future time points. If we did a preclinical trial like that, we would call that fraud. So please help me understand the validity of this method.
Great question. I think in general, all of these decisions about how to analyze the population, fall on a spectrum of conservativeness to less conservative, and they all can be fine in some situations. It really just depends on the context. So, last observation carried forward, the idea is basically all we know is what happened up until this point. And we are going to assume that, we're just going to take that as the last data point that we knew. Now, in some situation, you can imagine that if you're looking at a parameter that you're expecting you're looking at it to increase or decrease, for example, right? Like, we're looking at some scale, for example, or, something where we're expecting sort of an effect over time to change, then, that is a relatively conservative approach, right? It says, okay, in whatever parameter it is, let's say your hemoglobin A1C, okay, you started at 10, and now we're trying to reduce that. And you came back for your first visit at 9.7, and you came back for your next visit at 9.2, and then you dropped out, and then, you dropped out of the study, basically. Now, would you have continued trending downward, to more significant numbers? Well, of course, it seems plausible, but a very conservative thing to do there would be to say, look, let's just assume that you're a 9.2, because if we're still statistically significant, assuming, giving you your last observation carrying forward, then basically we have removed any possibility of bias. We've biased against a positive result. So if we still get a positive result, that's a good thing, basically. So I think that a lot of these decisions end up being around sort of where's the bias, right? Similarly, when you look, that's why the sort of intention to treat is the least biased way of analyzing clinical trial data. It basically says, fine, you have patients who are going to drop out because they get hit by a car, because they kill themselves, because they move to New Mexico, because whatever, all of those things can happen. And we're going to include all of them to avoid any possible suggestion that we're doing shenanigans here, right? To avoid any potential bias that we are introducing by doing anything other than an ITT.
Does this tie into the concept of censoring, which I've never quite understood?
Yeah, so censoring is a little bit, is basically a cousin of this question. So censoring comes up again in these time to event studies. So, again, in a time to event study, let's just take a cancer trial as an example. We're basically measuring survival, right? So the event is death, and we're going to track patients over time, and basically we're going to score you as dead if you die, and we're going to score you as, and we're going to continue to score you as not dead if you're still alive. Now, that's fine if every patient stays in the study. The problem is what happens if patients drop out. And when patient drops out, they become unevaluable for the endpoint, right? All we know, let's say the study was meant to run for a year and someone drops out after 3 months. We don't know how long they would have survived. All we know is that they did survive for those first 3 months, and that's it. So that's called a censored patient because they didn't have an event, but now they're kind of unevaluable for the, for whether they would have had an event. And the math of these time to event studies and Kaplan-Meier curves basically assumes that what it does is it basically allows you to estimate what would have happened to that patient based on the other patients who did progress past the 3-month time period. So every Kaplan-Meier curve you see, or every time to event study, includes some amount of estimation of what would have happened to the censored patients, which is legal as long as the censoring is what's called uninformative, which means that there's nothing there related to the actual efficacy of the drug. So I'll just give sort of a trivial example. If there's some side effect to the drug that actually is directly related to whether the drug is working or not working. So let's say, there are a bunch of drugs like in cancer, for example, where maybe you're going to get some bad skin rash, and the skin rash is directly related to the potency, right? So if you get a bad skin rash, it's probably going to work really well in you. And if you get a bad skin rash, it's not going to work that well. So in that case, if you drop out because you got a bad skin rash and you're just like, screw it, I don't want to be on this trial anymore, you're actually losing patients who probably would have been responders. More like, it's not uninformative anymore. It's actually informative censoring. So those are the kinds of situations that you want to watch out for in terms of censoring.
Yeah, I can see that would have come up with the Tarceva, which if you don't have the rash, it's not working.
Exactly.
Okay, so a couple of questions. The first one, this is about rare diseases analysis. I'm going to follow up with a question about adaptive design because we have a couple of questions with that. So here's the question of rare disease. How should we think about rare disease clinical trials that lack multiplicity analysis? Is this a common practice, and how should we interpret this?
Right. So there's a bunch of stuff baked in there. So,
you have 30 seconds.
Okay. So first, let's just tackle the multiplicity question, right? So multiplicity, the idea behind that is that if you test a bunch, stated simply, the idea is if I test a bunch of things in a clinical trial, and I leave all of them at a P of 0.05 level of significance, the odds that I'm going to get one of them showing up at P of less than 0.05 gets very high if I just keep testing a lot of different endpoints, basically, right? So, that's one reason why it's so important to be looking in clinicaltrials.gov and seeing, okay, what is really the primary endpoint versus what are secondary endpoints? I mean, let's just take a step back. The whole reason why FDA requires and why, we as readers want to know what's the primary endpoint is because in order to be kosher on the analysis, you have to pre-state what is the hypothesis that you are actually testing first and foremost. And if that doesn't work, you're not allowed to, go and say, okay, well, that didn't work out, but I have 50 other biomarkers that I'm going to test. And, now I'm going to report that this one ended up positive, and I'm not going to tell you about the other 49 that I tested that ended up being negative. So that's the multiplicity problem. I'm not sure that that's per se, that I think that that's a bigger issue in rare disease than in other types of things. I guess the one challenge that comes up a lot in rare disease studies is, two challenges come up. In some of these diseases, it's just not entirely clear what endpoint is that, whether there's a single endpoint. It hasn't been settled, like what's the real endpoint that you should be measuring. that is going to be important for clinical clinicians and regulators. So there's some amount of sort of exploring around that sometimes is required, and sometimes you end up picking wrong, right? Sometimes you've picked an endpoint in a study, and then you have as your primary endpoint, and then you have a secondary endpoint, which is also pretty important, but you just made a call to sort of subjugate that, put that down as a secondary endpoint, and the primary ends up being negative, and the secondary nominally ends up positive, even though that's not sort of kosher anymore statistically. I'm sure there are some other things I could think of, but I'm not sure that I don't have strong feelings about whether the multiplicity question is a particular issue with rare diseases.
Okay, your 30 seconds is up. We're going to move on now. I just wanted to show.
We should come back to rare diseases later, though, because I think there are some other interesting things to say about rare diseases.
Okay, adaptive trial design. Just quickly, my background changed because this is my favorite Kaplan-Meier curve. This was done for me by Jim Allison.
Okay.
So yes, from my collection now. Okay, adaptive trial design. We have when are adaptive Bayesian clinical trial designs appropriate? What type of patient populations or disease characteristics justify adaptive design?
Such a good question. I mean, I think adaptive trial religious adherents would say all the time, always, and forever. Realistically, there are a lot of really great advantages. First of all, I mean, let's just take a step back. Like, what are we talking about when we talk about adaptive trial design, right? Like, usually we're talking about something where the most common situation is where there's some interim look at the data done in a kind of ethically and sort of, appropriate manner that allows you to then make adjustments to the design of the study. And, that can take several different, that can be manifested in several different ways. So I'd say, some of the common things you see in adaptive trial designs are things like sample size re-estimation, for example, where you basically say, look, we're not exactly sure how big the effect is going to be. We're going to run the trial, and at some point in the middle, we're going to see how we're doing. And if the effect size looks like it's huge, we're going to kind of keep on trucking. If the effect size if it's clearly going to fail, we're going to stop the trial. And if we're in the middle, we're going to add patients, because now we know that the likely effect is going to be, is maybe a little bit smaller than what we anticipated. Now, you could have run that study and you could say the alternative there to running an adaptive trial would just be to run the bigger trial, right? You could have just said, let's say I'm deciding, should I run a 100-patient trial or a 250-patient trial? And that depends on sort of what difference you think you're going to see. It gets back to the powering question that we started at. And you could say, well, you should just run the 250-patient trial. The adaptive trial allows you to potentially save some money and also, save exposing a larger number of patients to a drug that doesn't work. So, if you can tell after 100 patients that the trial is going to fail or that the trial is going to succeed, then that's better than, than running it through 250 patients or some larger number of patients from an ethical point of view. There's also some financial and time-based issues that come into it, just in terms of sort of adding your, basically avoiding wasting money. There are some costs associated with sort of the setup and implementation of adaptive trials. I mean, these are non-trivial. And actually the whole thing about taking a look at the data in a kosher way requires a whole sort of data infrastructure, and there are vendors that specialize in this. But, in general, there are a lot of situations in which one could do an adaptive trial. And I think many more companies are sort of developing, continuing to develop the capabilities, et cetera. That being said, it's often just sort of simpler to design, spool up, and execute a non-adaptive study. So I think that's often, just from a logistical point of view, why companies just don't do adaptive studies for everything. That sometimes it's just not worth it.
I have a follow-up type of trial design that now it occurs to me, it might be just strictly academic, which is the umbrella trial. This is not umbrella. So the example I use is I-SPY, where I ran into this at least 15 years ago, and I was stunned that the regulatory actually allowed them to do it. So could you elaborate?
Yeah, I mean, I think there's a couple of, so first of all, I would put sort of basket, umbrella, and platform trials kind of all in the same category. They're all kind of different flavors of testing one or more drugs against one or more diseases and being able to adapt on the fly and sort of change the number of arms and change sort of how you allocate patients, right? I would say, the issue there ends up being, what are you using the trial for? Are you using it for initial hypothesis testing, or are you using it as a way to actually, for regulatory purposes, right? So I think for initial hypothesis testing, these types of studies make a lot of sense, right? And, both the basket and the umbrella. So, in one case, you're testing multiple drugs in one indication. I think that was the I-SPY situation, which was breast cancer, if I correctly. And I-SPY also had a component, if I remember correctly, where they were looking at biomarkers and then they were sort of adapting on the fly based on biomarkers. And then you could imagine the opposite, which is you have one drug and you're testing in a bunch of different indications. So this is the kind of thing that often happens in autoimmunity, for example, right? Is it going to work in Sjogren's syndrome? Is it going to work in, whatever, I'm just blanking on sort of random autoimmune disease, but whatever, lupus, et cetera, et cetera. You could say, okay, we're going to test it in all of them, and then we're going to sort of adapt on the fly. I think that, in a phase 2 study, those can often be really useful and important. In most cases, I don't think you would, you could use that as a lead-in to a registrational study. I'm not aware of any situations where someone has used that, submitted to regulators based on that type of trial. I think it's usually a prelude to running a more tightly constrained, trial, phase 3 study that tests a very specific hypothesis.
All right, now we have, again, this is all coming from chat. This is another type of trial, this is often definitely referred to in your book. Can we walk through a non-inferiority trial? And then 1, why would a company choose this route? 2, sure. Also, where the focus is lower bound margin in that study, how should one interpret a superior result? Parentheses, if the lower limit of the confidence interval is above the non-inferiority margin and above 0, and parentheses, is the chance of a superiority result in a non-inferiority trial unlikely because it's not powered to show?
Awesome. Okay, so what I would say a couple of things. First of all, we're not going to cover everything. We're not going to run a tutorial on non-inferiority trials in this context. There are a bunch of awesome open source reviews on non-inferiority studies. If you go into PubMed and you just look for reviews on non-inferiority studies, there are several of them that are open access, and I would definitely recommend them. They're all sort of, some of them will sort of cover in the context of specific therapeutic areas, but they all basically cover the same ground. So I'm going to defer. I'm using that as a way to sort of get out of, my get out of jail free card on running a stats tutorial. But I will say that the principle behind a non-inferiority study, and certainly the place in which it makes the most sense, is if you have a drug where the value proposition of the drug compared to an active comparator is not better efficacy, but some other benefit. So what could that benefit be? Well, for example, it could be a different route of administration. Let's say there's an IV drug, and now I've developed an oral form of that drug. And I'm not claiming that my oral or a drug that's comparable, right? And I'm making no claim that my oral drug is better than the IV drug. In fact, I might not even expect my oral drug to be better than the IV drug. But I do expect it to be in the ballpark of the same efficacy, but I think that it's going to have some other benefit. In this case, sort of a benefit to people who would rather take an oral drug than an IV drug, right? Or, a similar situation would happen in terms of safety and tolerability types of considerations, right? Like some sort of side effect. Like I have, someone's marketed a drug where, again, there's some bad rash side effect. And now I figured out a way to basically develop a drug that's very, very similar on the efficacy side, but
no rash.
But it doesn't cause the rash. And so I don't want to run an active control study proving superiority against the other drug, because I don't even think my drug is superior. I just think it's not worse. And those are the sort of classic situations in which a non-inferiority trial is entirely appropriate. Now, I mean, the question did raise this issue about, are there situations in which you can actually claim superiority off of a trial that was designed for non-inferiority. So it turns out, yes, those situations do exist. I would say they're relatively uncommon just because, again, usually the ingoing hypothesis for these studies is that the drugs are pretty similar. So really the biggest focus in these is missing the proof of non-inferiority, like actually showing you are inferior. That's the bad, that's the thing you really want to be watching out for in those studies. My sense is I'm not sure if I know of an example of an industry-sponsored non-inferiority study that actually then was able to claim superiority. It is a theoretical possibility. I'm just not sure that I can come up with an example off the top of my head.
Okay, again, we're going to switch gears here. There's a question here in chat. I'm going to fold my question in with it. My question has to do with cherry picking patient populations.
Awesome.
So you'll get this big slide, patient characteristics, males, females, blah, 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 blah. The question has to do with diversity and how do we address that in clinical trials. My question is more towards how can you look at this chart and see flags of cherry picking?
Okay. God, we're really hitting all the hits. This is an awesome, these are great questions. Okay, there are a couple things baked into that. So maybe I'm just gonna I'm gonna reorganize the question a little.
Yeah.
So, with any trial, with almost every trial, you're going to see a classic quote unquote table 1 reported, right? Which is the characteristics of the patients. And if it's a 2-arm trial, you're usually going to see the characteristics in each arm. So for example, how many males were there in the control? How many males were the experimental? How many people were, left-handed redheads in the control? How many were left-handed redheads in the experimental? How many had an albumin above X in the control, et cetera, right? So there are a couple of issues there. I think you raised this question of diversity in clinical trials. There's an ongoing problem. In fact, I just saw that there was an article in Stat today about sort of how hard it is, even when you're trying to make a very racially and ethnically diverse clinical trial, how hard it is in practice to actually get a racially and ethnically diverse clinical trial. I'm not an expert in clinical trials operations. I would say that this becomes, from an operational point of view, this becomes much more of a question, I think, for practitioners who are dealing with real live patients and trying to make treatment decisions, where really you're asking, okay, the patient who's sitting in front of me, how much do they or do they not resemble the patients who were studied in that trial? And how worried am I if it seems like they don't resemble it? If you're, African American or Asian, and this came up with the aducanumab studies, where the percentage of non-white patients was extremely low. And so the question is, okay, now you're dealing with a 70-year-old African American patient with dementia, and you're trying to put them on aducanumab, even if you believe the drug works, even if you thought the data were real, which we could talk about sort of later. The fact is that there were hardly any Black patients who were studied in that trial. And so you're left with a little bit of a problem in terms of assessing. I think in my world where I'm more, yes, that is something that one should be interested in, especially if you're interested in sort of commercial implications. But, I would say that rarely comes up in the sort of path to approval kind of discussions. It's mostly sort of a commercial, sort of how is it going to be used in the real world kind of question.
Okay.
I'll just sort of follow up quickly and say that the other thing, there were 2 other points that were raised there. Cherry picking to me is about subset analyses, right? So when I think about cherry picking, I think about, well, the bad news is our drug didn't work. The good news, it worked in left-handed redheads, with IQs above 140. And, using that as a rationale for, running another study or, doing more fundraising, et cetera, et cetera. That's extremely common. I would say, the short answer there to me is, this is about sort of assessing probability of success of sort of a next trial, right? So, really the question that you're asking is, okay, now the company is saying they want to run another study in left-handed redheads with IQs above 140. How de-risked what do I think the risk is of that trial, knowing that in a broader population, the trial did not succeed, but then it did in this post hoc analysis look like it worked in this subset. And I think that ends up boiling down to how confident you are in the original science, how logically chosen that hypothesis was, whether, about the left-handed redheads, whether there's any other ancillary information to make you believe that the story hangs together. Like if I were to say, okay, it looks like I got a signal in left-handed redheads, and also I have this biomarker that shows that actually the drug was 5 times more active, on this biomarker against in left-handed redheads. Okay, maybe I'd be interested. If there's no other supporting data and there's no biology to really, support the choice of that subset, then, and especially going back to the multiplicity question, if it seems to have been sort of randomly chosen from a large list of, tens or hundreds of different potential options, then obviously less interesting. And then I'll just make one other point, which is people get bogged down a lot of times on this whole table 1 thing about differences between the 2 arms, right? So oh, well, it looked like, the control arm had, people who were sicker or people who are, more males than females or whatever, and they'll find something. That's always a complicated situation. I will say that the practice of doing a p-value on those differences, on those parameters, has been widely debunked and sort of, it is not the right way to do things. Most top-tier journals don't let you do this anymore. There are still some second-tier journals that, that will allow this, but it doesn't mean anything for very statistical reasons we won't get into. I think the big issue there ends up being, on that parameter that looks like it's numerically different between the 2 arms, is that something that would plausibly bias the results of the results? And if so, in what direction? And that becomes a judgment call, honestly.
Okay, real quickly, if it is a semi-quick answer, you referred to the Alzheimer's drug that's of great controversy. A lot of people at the FDA actually quit when this drug was approved. Why did they quit?
Yeah, I mean, I think that this is, I'm not a regulatory guy. I know enough to be dangerous. I would say, there were a couple of issues there. When you think about just sort of how to evaluate late-stage trials, one of the reasons why the FDA generally requires 2 adequate, well-controlled studies was highlighted there by the aducanumab story, which is one of the studies, at least on the initial pass, looked statistically significant and the other did not. That generally is not, that generally precludes approval. So I think that's one strike against it from sort of a regulatory integrity standpoint. Second issue is that there were a lot of shenanigans played after the fact in terms of turning the negative study into a positive study and basically saying, well, we changed how many patients were allocated to the higher dose. And if we only look at those patients, then it does look like there was an effect. I'm not saying that those kinds of arguments are always don't hold water, but, from whether that's evidence to suggest that it is worth running a follow-up study is different from saying that that's enough evidence to suggest that the drug should be approved. And I think that's another place where people, took umbrage at the decision. And then the third was that, a lot of the most compelling data was basically surrogate endpoint data that had to do with levels of amyloid. That whole idea that amyloid levels are related to clinical outcomes, I don't think I'm the only person. This is going to come off strong, but I think that most people in the field believe that hypothesis has been largely debunked. There are obviously adherents, and we're not going to get into it. If you are one of those people, I apologize, and we can fight about it offline. But in general, again, giving a drug approval on a surrogate endpoint is something I think most people believe that FDA should be doing relatively sparingly. I think this is just the other thing that makes this just a really difficult situation is just the sheer size of the patient population. And it's clear that from a regulatory point of view, FDA views sort of the level of confidence that you need in a drug differently if you're going to be addressing, tens of millions of people versus if it's going to be, tens or hundreds or thousands of people, that there's just a different, and I think that there was a general view that that the agency used standards which might have been appropriate for an ultra rare disease, but used them to apply to a situation which is not a rare disease, and therefore felt like that was not appropriate.
Okay, I'm going to go back to one of my questions because I worked very hard on this. Morgen, can I have graphic number 5? And I'm hoping this is going to tie back into the rare disease that you just mentioned. Okay, what the hell is this? I have no idea. Kirk Schalper, who's a pathologist, he did this for me. I think they're T cells attacking something, but I'm not sure.
Yeah.
So this sort of ties into when I see especially preclinical stuff. And so we use mouse model XYZ, and this is what happened. And I'm like, well, that's a very nice looking mouse. Apparently it's not dead. This is all I can tell you about the mouse. You refer to this as intrinsic risk. There's something you just can't know. Does that sort of tilt into the rare disease space as well?
Yeah, I mean, again, this kind of gets beyond clinical trials, but I would say in general, well, I mean, the tie-in with clinical trials, I think is, in many situations that I find myself in, sort of, advising either investors or companies, you're looking at a trial which is either planned or ongoing and trying to figure out sort of what do you think is the probability of success. And I think there are a couple of components to that. And I mean, I would think I would outline 4, right? So one of them is what clinical data do we have? What clinical data led up to this study, right? So obviously, if something has been tested in people already, we have some amount of information, and what's the strength of that information? One piece of it would be, a second piece would be the design of that actual study, right? And that's some of the stuff we were talking about before. Some studies are designed in ways that are more or less likely to yield positive outcomes, either, dealing with power, dealing with the endpoints, et cetera, et cetera. Another category would be execution capabilities, right? So you may, we could, depending on the situation, you might believe that the same drug in the same indication, with the same trial design, with the study being run by Pfizer versus being run by 2 guys in a garage, may have better or worse probability of success in one of those situations. But I think the 4th piece is really what I would call what you refer to as the intrinsic scientific, the intrinsic likelihood from a science point of view, which is how good do you feel about the hypothesis, right? And that's where I think a lot of the preclinical information comes into play. I would say in the IO space specifically, you highlighted a problem, which I think is pretty common. And certainly I've had people tell me this, that a lot of the models that are being used are just too permissive. So in fact, I remember I was with a client once and we had gone to a major academic phase 1 center, basically trying to convince them to get psyched up about our study and run our trial. And we were showing them all of our mouse data. And the guy honestly was kind of ho-humming through all of it. And we went through 5, 6, 7 slides. And finally, I had it in a book and I closed the book and I put it down, face down on the table. And I said, look, here's the deal. If you tell us what preclinical experiment would get you excited about this, we will literally call it in from the train on the way back home to Boston, okay? Like, we will do whatever you tell us. If you say that there is a preclinical experiment that we could do that would change your view of the risk of this drug, of the likelihood that this drug will actually work. And the guy said, honestly, I can't tell you because there is no better model than what you're using. All of these models stink. And they're all just somewhat subjective. They're all just they add a halo of believability around the story, but they don't actually really move the needle. Now, obviously, there's a whole spectrum here in terms of diseases where I think if you look at certain disease areas, certainly some monogenic diseases where there are really good mouse models that recapitulate with high fidelity the clinical characteristics of the disease. Maybe you'd put those on one end of the spectrum, or actually think about infectious disease, where honestly the ability of a drug to kill the bug in a dish is a pretty good predictor of how good it is going to be at killing the bug in people. That might be at one end of sort of preclinical experiments that really are de-risking, and then the other end might be things where the models are largely derided as not being very significant at all. I remember I did a project last year in ischemic stroke, and there are a bunch of experiments that you can do in rats where you basically tie off a vessel and then you release the vessel, and you can look at sort of post-treatment. And basically, if you read, there are all these roundtables that have been done in this area. And essentially, most experts believe that those models are essentially worthless. They're nice for making pictures, but they don't actually de-risk the clinical development at all. And then you think about things like autism, other things in neuropsych land, I think where the models have typically been pretty awful. Things where it's clear that there are sort of fundamental anatomic or other type physiologic differences between animals and humans that make them just hard to model. So, I think everything's on that spectrum. And I think, at the end of the day, it's no use beating your head against it. I guess in my work, I find that either you're in a specific clinical area in which the models are the models, right? And you're just sort of doing the best you can. Or if you're an investor and you're looking across diseases, then sometimes it can be helpful to contextualize and say, okay, how good, all other things being equal, do I think that, the best preclinical data in autism is more or less de-risking than the best preclinical data in immuno-oncology versus the best preclinical data in antibacterials? That can sometimes be an interesting discussion on an investor level or on a portfolio level.
We just have a few minutes left. I'm going to ask a question. I'm going to ask a yes or no question. Does it matter where the trial is run geographically?
Absolutely, yes. For a couple of reasons. The most obvious of which is that standard of care can differ, between different regions of the world. So the kinds of drugs that people have been exposed to, sort of what are the characteristics of the patients who are going to go into the trial can depend greatly on what's available, what the technical capabilities are in that country, et cetera. I think we've seen that in some cancer studies where, you can run, if you are running a second line study or second line or more study, those might be very different patients depending on what, those patients may have different characteristics and have been exposed to different drugs. Depending on where you do it. And then of course, there's always differences in terms of the technical capabilities when you think about, especially in phase 2, but even in phase 3, if there are any issues in terms of how you assess the endpoint, you don't want it, you can end up with variability in terms of how the endpoint is assessed. That's one of the reasons why you often have centralized reading for some of these, for some types of.
Okay, we have a follow-up that feeds in directly to it. So here's the question. Have you seen any systematic comparison of investigator review versus central review? Because it seems like response rates and other outcomes always look better with investigator review and then worsen with a central review. So again, whose numbers do you trust?
Yeah, that's my sense as well. And I think the general feeling in the field is that central review is always preferable. Again, from our point of view, we're not designing trials where, you and I and the people on this call are probably analyzing trials. So I would say that when I read a trial that does not have centralized review, I just put that as another check mark in the potential bias / source of concern category. It's not that I automatically dismiss the study, but it's definitely something that I note.
All right, this one touches on something that I've been witness to any number of times, but again, I don't have the depth of knowledge to understand. You see so-and-so trial, arm A, arm B, whether arm B is the competitor is an active control or placebo. You look at the results and they say whatever they are, and then you come to your conclusion. However, the comparator arm in this trial did much better or much worse than one would expect. Without knowing all that history, there's no tip-off, right?
Yeah, no, I think it's a great point. I do think, one of the things I advise people when you're looking at a study, the very first things to do are really understand what the goals of the trial are and understand sort of the historical context. So, there are certainly, people sometimes refer to it as placebo effect. I think usually what people call placebo effect is really just high variability in either the clinical course of patients or in the measurement of the readout, right? So, you have a lot of diseases which are sort of waxing waning, or it's difficult to assess what the answer is. Like depression is the classic for this, right? Where, it's just, there's a lot of noise around, the control arm. That doesn't mean that there's placebo effect, just means that the result is noisy. Often
migraine is like that too. Migraine.
Yeah, exactly. So, I think that there are, a lot of these things that depend on patient-reported outcomes as as opposed to quote unquote, a more quote unquote objective outcome like a lab value that's measured by a physician. These are extremely fraught areas. And, I do think that it ends up, again, being a contextual problem where you have to decide, okay, what do you think actually happened here? And, I think the best clinical trials kind of take that variability and that potential into account and are already designed so that they are sized appropriately to, to deal with that, or they have other design elements. I mean, there's all sorts of other crazy designs you can do where you can sort of have patients switch between the two arms, and you have run-ins, you have all sorts of different ways of dealing with that. But absolutely, I think it's a very important, I think the take-home message there is it's very important to know when looking at a trial, if you're in a clinical area where there's been a lot of variability in how the placebo or control arm does.
I want to squeeze in one more question, if we can do it in like 30 seconds, which is press release shenanigans. This is the very first thing we often see. How do we spot the crap?
God, I don't know if I can do it in 30 seconds, but I'll
Give a couple examples.
I would say, any trial that does not start with a very clear, any press release that does not start with a very clear statement of, whether it was statistically significant on the primary outcome, anything that deviates from that is cause for concern, right? So once you start seeing stuff like, they mention an outcome, but it's not necessarily the primary outcome, or they mention that it was a subset of patients, or they mention that it was some sort of secondary analysis, like any of these sorts of things are basically, cause for alarm. Again, that doesn't necessarily, depending on the context, that's not necessarily, doesn't necessarily mean you throw out the baby with the bathwater, so to speak. I mean, in a phase 2 trial, you're going to see these studies all the time that are quote unquote nominally negative on a primary endpoint, and yet still justify further investment and could still, yield important information. But I think, the most upfront press releases are very upfront with what was the primary endpoint and did it meet it or not in the target population, and anything short of that is cause for alarm.
Okay, there we have it. We have been speaking with Frank David. He is the author of the Pharmagellan Guide to the Analyzing Biotech Clinical Trials. I want to perhaps take the prerogative here. This is the most questions I've ever seen typed in on chat. If you continue to do this, questions you did not get answered today, if we receive enough of them, perhaps Frank will come back.
Sure. No, I'd be happy to. I mean, I was just scanning through. There are a lot of awesome questions here. Yeah. Right. We could do this for hours. As long as
and we just might, but not today. So Morgen, take us out.
Yeah, thank you, Frank and Neil, so much. This has really been a very informative discussion, and we'll keep the listeners posted on when part 2 will occur.
Awesome. Looking forward. Thank you so much. Thanks, everyone.
Thank you.
Bye.